Ploidy refers to the number of chromosome sets in an organism. Let's say we have three types of chromosomes, A, B, and C. Therefore, the haploid number is 3. Now, there are two situations that are possible. The first situation is called euploid, where the number of different types of chromosomes is the same. For example, we might have 2 A's, 2 B's, and 2 C's, and this is the familiar diploid situation. Another euploid situation is when you have one of each type of chromosome, and this is called monoploid. And certain organisms such as male bees, wasps, and ants develop from unfertilized eggs in a process known as parthenogenesis, and therefore have only one copy of each chromosome, since fertilization was not required for their development. Now, you could have 3 of each chromosomes, or 4, and 3 sets that would be called triploid, 4 chromosomes of each type would be called tetraploid, and so on. And one could write this in terms of the haploid number of chromosomes n. So this would be 2n, n, 3n, 4n, and so on. And having more than 2, this situation is called polyploid. Now, it's also possible in some situations where you have a differing number of chromosomes. For instance, I could have just one copy of the A chromosome while having 2 copies of B and C, or I could have 2 A's and 1 B, 2 C's, 2 A's, 2 B's, and 1 C. This situation, the number of chromosomes we have is 1 less than the diploid, so it's 2n minus 1, and this is known as monosomic or monosomy. You could have more than 2 chromosomes, so 3 chromosomes for 1 chromosome, while having the normal diploid number for the remaining chromosomes. For instance, you could have 3 A's, 2 B's, and 2 C's, or you could have 2 A's, 3 B's, and 2 C's, or 2 A's, 2 B's, and 3 C's. Here we have 1 extra chromosome over the diploid number, so we have 2n plus 1 chromosomes, and this is known as trisomy or trisomic. Now, polyploidy has been especially common in plants, and it has played a major role in the evolution of new species in plants. And one piece of evidence that we have that shows that polyploidy is involved in the evolution of plants is that after the haploid chromosome number 12, even number of chromosomes are much more common in plants than odd number of chromosomes, which suggests that many plant species evolved because of a spontaneous doubling of chromosomes. One interesting effect that polyploidy has on plants is that it causes them to grow bigger. For example, the leaf and flowers of a diploid watermelon are shown on the left, whereas the leaves and flowers of a tetraploid watermelon are shown on the right, and it's obvious that the tetraploid has bigger leaves and flowers and bigger fruit, and that's why creating polyploids or harnessing naturally occurring polyploids is important to agriculture, because it increases crop yields. So let's consider how polyploid organisms, plants or animals, but mostly plants, form. This could occur naturally in the wild, or could be carried out on purpose as part of agriculture. Let's say we have a tetraploid species and we cross it to a diploid species. The gametes of the tetraploid are going to be 2n, and the gametes of the diploid are haploid, so they are n. And if we cross them, then the hybrid would have 3n chromosomes, so they would be a triploid. Now, as it happens, triploids are usually sterile. And in order to understand why triploids are sterile, let's look at meiosis in triploids. So let's say we are looking at meiosis 1 during prophase. There are two possible ways that 3 chromosomes, in this case they would be dyads, you would have 3 pairs of sister chromatids, and there's 2 different ways that they could pair up or synapse. One possibility is that 2 pair up, so that's a bivalent, and the third one does not pair up, and that's a univalent. The second way this could happen is that all 3 pair up, forming a trivalent. During anaphase, when these chromatids are being pulled apart, when you have a bivalent, they will move to opposite sides, opposite poles. On the other hand, the univalent could go to either pole, and the choice is made randomly. Irrespective of the choice the univalent makes, in segregating, we will always end up with 2 chromatids on one side of the metaphase plate and 1 chromatid on the other side of the metaphase plate, resulting in 1 haploid gamete and 1 diploid gamete. Now, when you look at the trivalent arrangement, where 3 chromatids have synapsed, there is always a pair that have paired up, and they segregate to opposite ends of the pole. And the third one, just like in the univalent case, could go to either side, and the choice is made randomly. Once again, as in the previous example of a bivalent and a univalent pairing, we will end up with 2 chromatids on one side of the metaphase plate and 1 chromatid on the other side of the metaphase plate, resulting in 1 haploid gamete and one diploid gamete. So let's extrapolate this to the situation where we have two types of chromosomes. So it's triploid, and so we have triplets of two chromosomes. Let's say one is blue, and we will end up with two blues on one side and one blue on the other side. And let's say we have red chromosomes, and we could end up with two reds on this side and one red on the other. When meiosis has completed, this will result in two types of gametes. You would have one red and two blues, and so this gamete would be diploid for the blue chromosomes and haploid for the red chromosome. Or you could have, from this side, one blue chromosome and two red chromosomes. Of course, since these chromosomes assort independently and randomly, another possibility of the assortment is having the two reds and the two blues on the same side, and in that situation we will get gametes with two reds and two blues. Therefore, this gamete would be diploid for both the red and the blue chromosomes, and one red and one blue chromosome in the gamete. So the gamete is haploid for both the chromosomes. Now, it's quite unlikely to get such gametes that are haploid for both the chromosomes. To see that, first we note that 50% of the gametes will be diploid for a given chromosome, and 50% will be haploid. Therefore, the probability of getting one chromosome, so being haploid for the blue, is half, and the probability of being haploid for the red chromosome is also half. And in order to get both being haploid, we use the product rule, and the probability of getting haploid for two chromosomes is going to be a half times half. And that can be seen here because we have a 1 out of the 4 different gametes is haploid for both the chromosomes. Now we can extrapolate this to say that if our haploid number is n, then the probability of getting all haploid is going to be a half times half n times. Which is equal to 1 over 2 to the power n. Now, if let's say we have a haploid number similar to humans, n is 23, then the probability that you will get an all haploid gamete is going to be 1 over 2 to the power 23, which is something like 1 in 8 million. This is also true for this situation, where you get gametes that are diploid for every chromosome. So the probability of all being diploid is also 1 over 2 to the power n, which is a very small number. And these two situations, where you have either all haploid or all diploid, they are euploid, where the chromosome number is the same for all the chromosomes. As a result, most of the gametes are going to be aneuploid, and that's the reason why triploids are usually sterile. Most of the gametes are aneuploid, so that the zygote ends up being aneuploid. And aneuploidy is usually lethal, since you have either too small a dosage of many genes, or too much of a dosage of many genes, the genes that reside on the aneuploid chromosomes.